Hello, everybody. We're once again in the section What We're Reading Today of the South American Mycorrhizal Network. And today we're with Isabel Mujica. Hello, Isa. How are you?
Hi, fine, thank you. And you, Guille?
Fine, fine, given the circumstances.
Yes,
So
you're
Isa
right.
has been working a lot on orchid mycorrhizal fungi and has published recently 2 papers. Be- So let's start. Before going to the papers, I want to ask you, why are you interested in orchid mycorrhiza?
Well, I first of all thank you, Guille, for the invitation. It's an honor to me for me. Well, orchid mycorrhiza it is a great model study to study symbiosis. Uh, the difference between this type of mycorrhiza and the other type of mycorrhiza is that here the fungal partners also participate in the germination of seeds of orchids. So the orchids have very small seeds without energetic reserve, so they need the assistance of these fungal partners to germinate. So it is not only a relationship between adult plants and the fungi, but also during the germination. So orchids have a really high dependence on this symbiosis. And this, I think, is very interesting to study, the evolution and the ecology of these interactions.
Very
Yes.
interesting. I noticed that actually in 3 of your papers you use the same genus and sometimes the same species, Bibinola fimbriata. Why do you find this
Yes.
a perfect model? Why so?
Yes. No, I don't know if a perfect model, but at least in Chile, this is one of the most widespread orchid. Bibinola fimbriata forms very extensive populations and very dense, so you can find it from the north of Chile to some central south. So it is very, it has a very wide range of environmental conditions. So it allows us to test a lot of hypotheses. And it is very easy to cultivate, and we already have worked with this species, so we know more things and we can advance in theory with Bipinula.
Cool, cool, cool.
Yes, and it's very
One.
beautiful.
So what is the? Let's focus on the first paper about soil phosphorus. And so what is this paper about?
Well, we in a past study, we saw that there was a relationship between nutrients, soil nutrients, and the diversity of fungi that we found in roots of these orchids, of Bibinola fimbriata. And we saw that with more nutrients, the orchid presented more specialized interactions, less, lower diversity of fungi. But this was a very correlational study because we study a lot of populations and we saw the relationship, the correlation, but we wanted to do something more experimental. So adding, in this case, phosphorus, and see what happened in the roots of this Bipinula. And also in the other study, we mostly investigate the diversity of orchid mycorrhizal fungi, not all the fungi that is in the roots. And now we observe all the diversity using meta-sequencing. So in this case, we were able to see the effect of nutrients in all the community, the fungal community, including mycorrhizal fungi, pathogens, saprophytic fungi, etc. So that was the idea of this study.
And this study is focused on one population, one big population, and you have 4 different locations within that population that is reflected in this figure that you have, and then you show the percentage of each different fungal guild. And one thing that it's kind of surprising is that in the south you have more pathogens than mycorrhiza, mycorrhizal fungi. And I wonder what can drive this pattern. It can be the salinity because it's surrounded by the sea, and I wonder if the wind brings some I don't know, some salt or moisture, or what do you think will be the
Yes.
driver?
Yes, this one was one thing that was very surprising because this is, as you just said, it was only one population, relatively small, and it is very structured. So the south is very different from the northern side of this. It is a peninsula. And one thing that is very different is the humidity, because in the north you have more exposure to sun, and the soils are more dry than the south side of this peninsula. So this can be one factor. And also the phosphorus, this is what we discuss in the paper, it is not homogeneous distributed, because we have in one side a colony of birds, of seabirds, that it is a big input of phosphorus into this site.
Yeah.
And this is very interesting also because this produce that inside this one population we have a very wide rank of phosphorus content in soil, from very high points that were very high to others that were very low of phosphorus.
One thing in that same case, in this different aspect, you found that in the south, where you have less mycorrhizal orchid mycorrhizal fungi, you have more pathogens, but then you still have some orchid mycorrhizal fungi. And I was wondering if you think it is possible that this survival mycorrhizal fungi there can have other functions, like protection against pathogens, for instance.
Yes. Well, one of the main results of this study is that when we put phosphorus, this produced a decrease in mycorrhizal colonization, and this had a consequence in the richness of pathogens. Pathogen richness increased when colonization decreases. So this is something that is telling us that possibly the orchid mycorrhizal fungi has a function against pathogens. But it is interesting to think that within the community of orchid mycorrhizal fungi, there might be a variation in this function of defense function. And also one thing that was remarkably was that in another study that appears the same year with Bibinola fimbriata, a research group were studying the gene expression of roots in Bibinola fimbriata, and they found that colonized roots with mycorrhizal fungi expressed more genes against pathogens than not colonized roots.
Very
so
interesting.
there, and I can send you this reference if someone wants to read it, because there is the mechanism that might be explaining what we saw in this experiment. So it's really something is happening in defense against pathogens in orchid mycorrhiza. Yes.
So I think that's actually the main message of the paper, I think, right?
Yes. It's also that inside the roots there is a fungal community, okay, with different guilds of fungi, and they are interacting within the roots. And if you modify the environment in which this plant is living, the relationships within the roots change. And that is something new that has not been studied in orchids before. So yes, that is some novelty about our paper.
Yeah, that's really, I like it very much. I think this is also related to your second paper, where you focus more on nutrients and fungal identity, and you isolate the fungi. So let's move to the second paper, which is entitled nutrients and fungal identity affect the outcome of symbiotic germination in Bibinola fimbriata. So what is this paper about?
So all these papers are trying to understand the effect of nutrients in the symbiosis between orchids and mycorrhizal fungi. And the one that we just discussed was about adult plants in situ, in the nature, trying to find the effect of phosphorus. But as we talk in the beginning of the talk, the mycorrhizal fungi in orchids have a really crucial role during germination. So it is possible that this effect of nutrients that we saw in nature is because in that stage, during germination, there is an effect of nutrients that is combined with the identity of fungi. So we wanted, with this second paper, we want to evaluate that aspect of the ecology of orchids. So in this article, in this experiment, we have different fungal strains of Tulasnella and Ceratobasidium. These are genera very common in orchid mycorrhizal fungi, and different conditions of nutrients. And we wanted to see how what was the effect in one side of the identity of mycorrhizal fungi and the addition of nutrients on symbiotic germination. Yes.
I'm going to ask you what will be the result, but before I have a technical question. That in the first paper you found that the OMF, the fungi more abundant in the roots of Bibinola was many groups, but before was Ceratobasidiaceae and then Sebacinales and Tulasnellaceae. But in this one, you use the first one and the third, Ceratobasidiaceae and Tulasnellaceae. And I wonder what happened with the Sebacinales. Is there any issue?
It's a very good question, because these both studies we did it in parallel. So we have the isolates, the fungal strains from the past study that was about Bibinola in different populations. So, and there we found that Bibinola only associate with Tulasnella and Ceratobasidium, because this study was, we evaluate the diversity by culturing the fungi. And we never found Serendipitaceae, that is the other family that form orchid mycorrhizal fungi. So then we have the results of the study of Zapallar that was doing next generation sequencing, and we found Serendipitaceae, but we didn't know that it was associated with Bibinola. And the other thing is that in all studies that have been made in Chile on orchid mycorrhizal fungi, they never have found Serendipitaceae. So we thought that it was not there. So we didn't include it.
You discovered that you find it in the first paper.
We discovered, and we don't have the isolates. So yes, that's why.
So what are the main results? What is the main message of this second paper?
Well, in this second paper, we found that nutrients have an effect on the outcome of the germination, but it depends on the identity of the mycorrhizal fungi. So in some of these fungi, specifically Ceratobasidium, 2 species, we put more nutrients and the mycorrhizal fungi still germinate the seeds. So it was mutualistic all the time. Despite we put more nitrogen and phosphorus, the fungi still was germinating the seeds. But contrary, the other isolates that were other species of Ceratobasidium and Tulasnella, the time we put more nutrients, they became pathogens. They didn't germinate the seeds, and they grew over the seeds without being symbiotic. So this is telling us that there is a lot of difference in the response to nutrients depending on the mycorrhizal fungi. And if we do some speculations, if we think in one site with a lot of nutrients, as the ones we saw in the study in this peninsula, there will be only a few fungi, a few species that will be able to germinate the seeds. Contrary, in places with low concentrations of nutrients, there will be a lot of mutualistic partners. And this will result in populations with a higher diversity of mycorrhizal fungi, as we saw in the other study in Bibinola.
Oh, very interesting.
So it is, yes, and the thing that is more surprising to me is that there is another study that has just been published in Mycorrhiza, in the journal Mycorrhiza, by Thomas Figura and other colleagues, and they found in European orchids, terrestrial orchids, they found the same pattern, that only with some species of Ceratobasidium, there is no effect of nitrate in this case. They try only with nitrogen, and there is no effect. But in the other fungi, you put some nitrogen and there is no germination. So we want to see if maybe other orchids, maybe all orchids have this pattern. We are just starting with this. Yes, it's.
No, very, very, very interesting. So it's just where you're developing or showing the complexity depending on the nutrient and on the fungi. So it's more complex that.
Yes, because there can be a species, a fungal species, that you say, oh, this species is very mutualistic. It always germinates the seeds. It is very fast to germinate. But you change the environmental conditions, you put some nutrient more, and there is no, yes, there is no interaction, or there is a pathogenic response.
Very, very interesting. So what do you think will be the next step? What would be the most immediate future for you or for orchid mycorrhizal research? What will be interesting to do next?
Well, in this thing that we were talking recently, I think that another aspect that should be considered is the biotic interaction, because we have the identity of the fungi, the abiotic conditions that can be nutrients. There are other studies with light or carbon source, but also the biotic interactions. For example, what happens if you have a community of fungi and you change the conditions? Maybe one fungal species start to dominate and become the only one in the community, or I don't know, there is more competition. Yes. Well, we have a student that is doing something about it. So we will see what happened with it's like how competition between mycorrhizal fungi affect the outcome of the germination. Yes, I think that is
very interesting.
Yes.
Good. Thanks a lot, Isa. Very interesting papers.
Yeah. And if anyone has some questions or want to know more or want to collaborate, they can write to me. I will be happy to.
Yeah, we can add your email in the post so people can write you if they feel like.
Yes, of course. Thank you. Thank you very much.
Thank you very much. So bye bye, Isa.
Bye bye.